Good afternoon, everybody. Welcome to the latest webinar by MBF Bioscience. I'm Sue Tappan, staff scientist here at MBF. I also run the day-to-day operations at our contract stereology research facility, MBF Labs. I'm joined by Dr. Dara Dickstein, an assistant professor at the Icahn School of Medicine at Mount Sinai. Today we're going to be discussing how to prepare your tissue with automated reconstruction in mind. In addition, I'll be showing off our latest software for automated neuronal reconstruction, Neurolucida 360. I've had the pleasure of working with Dara for the past few years on this particular project, and I'm thrilled to be presenting this webinar with her today. As I mentioned, Dr. Dickstein is an assistant professor at Icahn School of Medicine at Mount Sinai. She received her PhD in genetics from the University of British Columbia, looking at the role of inflammation in Alzheimer's disease. She then moved to New York to do a postdoc at Mount Sinai, working in the field of aging and Alzheimer's, in particular the morphological changes of normal aging and disease progression. That's her on the left side of your screen, while I'm there on your right. We both have a shared love of the Pacific Northwest. I did a postdoc at the University of Washington and would go up to Vancouver to watch my favorite hockey team, the Vancouver Canucks, play on occasion. Dara, this is a stunning picture. Where's this image from?
This is taken in Revelstoke, British Columbia, and it's the highest skiable vertical in North America. It's awesome.
So you took this picture?
I did, and it was a great
Oh,
powder day too, so.
well, yeah, look at that sun. It looks fantastic. I'm really pleased that you took time out of your busy schedule to join me today. And I want to thank everybody who's online right now for coming as well. Let's take a few minutes to cover some of the basic information about the webinar before we get into the discussion on automated reconstruction. So this webinar is being recorded and will be available to view later. A link to the webinar will be sent to you in an email. If you'd like a certificate of completion for attending this webinar, just let us know in the question window and we'll provide one to you. All questions and answers asked during the webinar will be available on our website, even if Dara and I don't get a chance to answer them live. Dara, why don't you get us started?
Okay, so I guess the overall reason for doing this seminar is why assess neuron and spine morphology? Why do we care about it? And we know in development and in aging, which is what I'm familiar with, that we have significant changes in neuronal structure and spine densities and structures with aging. And just here you can see from macaque monkeys, the left being young, the right being old, there's significant changes in neuron morphology and complexity and length with age. And above, you can see, again, significant changes in spine density and also spine type with a loss of thin spines with aging. In other situations such as stress, we also know that there's significant changes in neuronal length and complexity when exposed to stress, and these can recover over time. And in addition to just changes in the neurons, there's also changes in spine density and again in spine type. If we look into neurodegenerative diseases, which is my field of expertise, we can see that in an Alzheimer's disease model, on the next slide, which has a mutation in the tau protein, you can see significant neuronal atrophy with disease progression. And in addition to neuronal atrophy, we see significant decreases in the spine density, and not only in the density of the spines, but in the types of the spines. And this is really important when you're trying to, um, you know, explain the cognitive decline going on in these diseases. We have this decrease in mushroom spines, which are thought to be the stable memory spines. In other models, such as the model with amyloid precursor protein, again, you can see there's changes in neuronal atrophy, neuronal complexity. And in this situation, we didn't necessarily see a significant decrease in the spine type. But what was interesting is there was a decrease in the spine density, but we saw a decrease in the spine type, where there are changes in the mushroom spines again. And so this really shows that it's really crucial and important to be able to reconstruct neurons in 3D accurately, and more so reconstruct the spines in 3D accurately, so you can categorize them according to their shape because they do have different functions. There are three types of spines that have been characterized, and they're mushroom spines, which are rich in AMPA receptors. And as I mentioned before, they seem to be these more stable synapses and are thought to be more involved with memory and memory spines. And these are usually the ones that are lost in neurodegenerative disorders. There's also the thin spines, which have the fewer AMPA receptors and more plastic, and they're thought to be the more learning spines and are really high in number in development. And then there's the stubby spine, which again, this spine has no neck to it. It's just a protrusion coming from the dendrite, and it has a free diffusion of molecules. And the function of the stubby spine is still debated in the field. There's been changes in stubby spines with stress, with depression, but again, it's still not 100% known the true function of the stubby spine. And for a good review on spine structure, spine function, Kristen Harris has many reviews from over the years that people can look up. In particular, there's one from 1994 with Kristen Harris and Cater that is a really nice review of spines. So given that, we know we have these biological questions, we want to look at morphology changes in full neurons and in spines. How do we label our neurons? How do you label these cells? And I'm just going to go through a few um, I'm just going to go through a few examples of how to do this on the next slide. And so there's ionophoretic injections of fluorescent dye, which I'm going to refer to as cell loading from now on because it's a bit of a mouthful. There's cell filling in recorded neurons, GFP labeled cells in transgenics, or so on and so forth, and dialystic labeling with lipophilic dyes. So for cell loading, and this is near and dear to my heart since this is what we really do in my laboratory, it's an injection of a fluorescent dye. We use Lucifer yellow, but you can use any other Alexa Fluor fluorescent dyes as well. And in this method, the perfusion is critical. If you have a bad perfusion, you will not get a nice loaded cell. And we do a flush with a 1% paraformaldehyde solution, followed by 4% paraformaldehyde with a 0.125% glutaraldehyde. It's important that in this situation, and in most perfusion situations, that you have fresh made paraformaldehyde with a pH between 7.2 and 7.4. This is critical for a good perfusion. And once you have your tissue and you fix it over 6 hours to overnight, we then cut it on a vibratome. And based on studies from many other labs and labs here at Mount Sinai and other labs, we found that for a mouse, we cut at 200 microns, a rat 250 microns, and a monkey at 400 microns. And we find that this thickness will allow for full neuronal filling in 3D of all these cells. We'll get really nice branches without very many incomplete or cut branches. So you really get a nice representation of a neuron. And then we actually keep this on PBS with azide, and we found that if you do this, you can come back to these sections for years and years after you actually start or you load your cells. Another important thing about in any situation when you are mounting your cells on your slides, we actually use spacers now that you can get from EMS or Fisher Scientific. And these spacers allow, they're about 100 microns thick, and if you usually just put your tissue on a slide and put a cover slip and then seal it with nail polish, you can actually get smooshing, or the scientific word, or compression of your tissue, which can then affect the visualization when it comes to imaging because it's a bit flattened and it can change the spine structure, dendritic shape. And so we use, depending on the tissue, 2, 3, 4 spacers, one on top of another, and then we cover slip it. And this also provides a nice tight seal so you can come back to your tissue years later if there's no air bubbles and it doesn't dry out. Another thing that's important about mounting is the cover slip that you use. Every microscope, the objectives are designed to use a 1.5 cover slip. And some people will use a 1 cover slip just to have a bigger working distance, but then this is going to create a bit more aberrations in the Z plane or spherical aberrations when you're doing your imaging, which can be corrected post imaging with different softwares, but can't always be corrected. So it's really important to use the proper cover slip. So to kind of go back to the cell loading on the next slide, we can see that what's important in this technique is that you evenly space your cells far enough apart. And here's a beautiful image from John Morrison's group at Mount Sinai showing neurons in a macaque monkey spaced apart so that there's no overlap in dendritic segments. It's easy to trace. When it comes to doing a full neuron reconstruction or full neuron imaging, there's no interference from any other cell. It's all, as I mentioned before, it's dependent on the perfusion. It's important you don't overfix the tissue and you don't overfill the tissue, or else you'll have a lot of leakiness and some ghost cells around it, which again will make it more difficult to reconstruct in 3D. Another great thing about this technique is if you're using GFP labeled cells and there's a bit and the signal's a little weak, you can then inject those GFP cells with a lucifer yellow, any other color dye, to get some enhanced signal. Or if you're doing some sort of retrograde labeling of cells and you don't get a full filling of those cells from the tracer, you can then use this technique to inject those retrograde labeled cells and get full reconstructions.
So, Dara, can I ask a couple of quick questions?
Sure.
When you're choosing these cells to label, to inject, you're looking to find those somas that are within the depth of that tissue section, right? So you're cutting those slabs at 200 to 400 microns, depending on the species. But then you're looking for the somas sort of in the middle of the section thickness.
Right. You don't want to inject the somas that are closest to the surface because you will get incomplete filling, meaning you're going to get a lot of cut dendrites. You don't want to you don't want to aim for the cells that are really too deep in the tissue because then you're going to have your neurons will be too deep and then you'll go beyond the working distance of any microscope to do the reconstruction. You kind of want to get the cells that come into view second. And those ones, if you inject those ones, you really will get nice dendritic trees, minimal abrupt or cut branches. I mean, that can happen in anything because it's a 3D structure within the good working distance of your objectives on your microscope when you start imaging.
So that's where that sweet spot of the section thickness really comes in, is that it's sort of a trade-off between the arbor extent that you might be looking for the cell type that you're interested in, as well as the working distance of your microscope and what you're setting up for that.
Right. So in addition to the cell loading technique, there are other techniques we can do. Self-filling in recorded neurons. So there's quite a few people who do electrophysiology recordings. And it's really nice that once you do have your cell clamped, you can simultaneously fill with 1% biocytin. And I think this is really strong and powerful because now you're going to have information from a recorded neuron. And now you're going to have morphology information based on the recording. So I think that's really strong and really, really powerful data. And again, you just simultaneously fill the neuron as you're recording and then take your slice, fix it in paraformaldehyde, and then visualize your cells by doing an immunohistochemistry with streptavidin and Alexa antibody. And as you can see here, this is a neuron that we have from a macaque monkey that's been electrophysiologically recorded and then filled with biocytin. And here we did a montage of multiple panels to get the entire neuron in our field of view. And you can see how beautiful this neuron has filled. You can see apical dendrites, the basal dendrites, and even the axon coming up with spines on this as well. So it's a really great and very powerful method of loading neurons in your cells. In addition, there's GFP, and many labs are now having GFP mice crossing it with their disease mouse of interest, and you really do get quite beautiful images of neurons. And as you can see in the top left panel, there's this whole sea of neurons. And while that's really nice, it's really a little difficult to reconstruct all of that. Because you will have a lot of overlapping dendritic segments, whether they are far enough in the Z or if they're close in the Z, that really could have a problem with reconstructing. You can also do virally expressed GFP in specific cell populations. So if you want to focus, say, on the nucleus accumbens or the amygdala, you can really focus in on there, and that's really great and easy to reconstruct in that respect. If you want to get a bit more sophisticated, you can look in a Crelox transgenic mouse system. You can look at CRISPR GFP expression and using also the Brainbow mouse, which Susan will talk about a little later on. So a couple of the downfalls with this could be what I just mentioned, that you get so many cells and it's a little bit difficult to reconstruct. You can also get some weak expression of your GFP, but that can be fixed and by just enhancing the signal with an antibody against GFP. And finally, as I mentioned before, you can also fill GFP labeled cells with dyes before. But as you can see in the top left panel, you can get some beautiful, the top panels in the middle and then the far right, you can get some really beautiful neurons and beautiful segments with nice defined spines, which is really great and great for imaging.
I really like this last point that you mentioned, where you can have your transgenic mouse with GFP under a Thy1 promoter, and then use that as your roadmap to come back in and select cells to fill with another dye, maybe a different color,
Yeah.
so that you can use it for targeting, which I think is fabulous.
Definitely. So finally, the last way we can think about loading cells is using dialystics. And when I started my postdoc, I started trying to optimize this technique in the lab, and it was a little tricky, I have to admit. And this uses the gene gun, the Biorad gene gun, to introduce lipophilic dye into cells, which gets incorporated into the cytoplasm and plasma membrane. And gene gun was originally used to do transfection of DNA in culture, and it's just been adjusted to do this. Again, it's really dependent on the perfusion and the fixation time, in particular the fixation time here. But there are some caveats to this technique, and you can see that you're not necessarily focusing on one individual neuron like the previous methods. Here you're shooting your tissue with this gun, with these tungsten coated bullets with fluorescent dye. And as you can see, you get a lot of these little bullet um, labeling throughout the tissue. Um, you can get a lot of undefined cells. So you're going to have little random dendrites here and there, and you don't really know, is that an apical dendrite? Is that a basal dendrite? And if you want to distinguish between the basal and apical, it's a bit more difficult. You also don't necessarily get really defined somas, and when it comes to 3D reconstruction of this, it's very difficult to follow through. And as you can see on the bottom um, left, this is an example of what a piece of your tissue is going to look like. And while you're focusing on the CA1, and they did get some beautiful neurons in this study, your yield of neurons is going to be low per animal. And when we do these studies normally, we like to do, power analyses have shown to get good statistical power, you need 5 to 10 animals per group, 5 neurons per animal, at least 5 to 10 neurons per animal. And then if you're doing dendritic segments like we do for our spine analyses, we like to get at least 3 to 5 segments per neuron per mouse. So it's a lot.
Let's go over that math again. So
Yeah.
you're talking, let's see, so 3 segments per neuron, five 5 to 10 neurons per animal.
So at least 15 segments per animal.
Okay, so 15 segments per animal, 5 to 10 animals per
Group.
group, and then and this is just at the tissue prep stage, and then you got to carry all of those steps all the way through to analysis.
Yes.
So,
So it's not a quick technique, but
well, it's true for any of these techniques,
yeah.
right? It's not specific to dialytic.
No, that's applied across the board to whether you're doing cell loading, filling of recorded neurons, or GFP neurons. I mean, we've done enough power analyses and published enough to show that's kind of the minimum number of samples that you really do need to get good statistical significance.
Right. So an automated method for reconstructing those neurons and modeling those spines may be of interest to not just you, but a few other folks as well.
Well, it'll make things go much faster as opposed to if you have to trace manually and do all the spine counting manually, which is what I did when I first started here, which took forever. But yeah, no, having an automated software to do that will definitely save so much time.
Leaving you to have time to, I don't know, do more science.
True.
So the next step then is once you have your tissue, you've got to digitize it. You got to get some good images.
Right. So you have to image your tissue, and this is a big problem I've seen in the field. And when I see some papers and they show these oversaturated images of neurons and dendrites, and they tell me a spine density, and I kind of look and I think, how do you know? Because you've oversaturated your image, and I don't necessarily believe the data there. So I think what's really important to image is first you need to select the magnification based on what you're imaging. Are you imaging a neuron? Are you imaging dendritic segments for spines? Do you want your neuron to have the spines on it that you can quantify or no? So if you're just looking at wanting to do like a neuron reconstruction only, you can use a lower magnification. I would still use a high resolution, but if you want that one neuron in your field of view, you can use a lower magnification. I would still, in a lot of instances, if you're looking at CA1 neurons, you're not going to be able to fit a full neuron in a 40x magnification. So you're going to have to do a tiling effect. And again, that would just give you better resolution too. And I think it's so much better for your reconstruction. So once you figure out what it is you want to image and the magnification you want to image at, you then want to figure out what your resolution is. And we tend to do a high resolution. And for us, we find that 512 by 512 is sufficient. You can go higher. I wouldn't necessarily recommend going lower than this. Um, Another thing that's important is correct for your refractive index. And here you're going from, like, if you think of how your laser is going in the microscope, you're going from air to oil to an aqueous mounting media to your specimen, and you're going to have all this aberration in the Z as a result when you get your image. And so you really should correct for your refractive index. And any mounting media you have will have a little number that says what the refractive index is, and then you can correct it accordingly on the microscope you're using.
What mounting media do you typically use?
We use a couple. We use Fluoramount G. Um, there's a Vector Shield, but you have to make sure you're not going to use a hard set mounting media. You want to use one that will always stay, your specimen won't get hard. Um, but there's quite a few out there. You can use ones that also will prevent fading as well. There's a few out there that have that. Uh, but we usually use Fluoramount G. Once you've kind of set up your microscope, now you want to look at your specimen and you want to focus through it and figure out your offset and your gain. And this is incredibly important. And so if you look on the image on the screen here where the yellow arrow is pointing to, you can see that there are these two spines coming out at you in the Z plane. And if this image was super saturated, so if we just say false color this as red and blue, and red being our signal and blue being this background, I've seen some instances where, people will have the entire dendrite red. And when it comes time to do the analysis, you're going to lose those spines in that Z plane. I mean, right from this angle, I don't know if that's a stubby or if it's a mushroom or if it's a thin. And if you supersaturate your dendrite, you might actually lose that information. So what it could be a thin or a mushroom might be quantified as a stubby because you've lost the neck. You have to make sure that you really don't oversaturate when you set your offset and your gain. Same thing as when you're taking, the image, you don't want to get rid of all of your background because if you think of it, the smallest total volume of a spine is 0.04 um, for your head volume, and that's really small. And if you're taking away all of your background noise, you can actually be taking away a lot of signal, which could be um, the spine head. And so you can again alter and bias your data that way as well. So when people ask me what kind of picture to take, I say take an ugly picture. Don't take a picture that you want to have published.
Right. I say that also for other applications like stereology. It's important that you have the
Yeah.
information you need. It doesn't have to be the the cleanest or most stunning image. You can go back and collect that image in addition, but that's not the one you necessarily want to quantify from.
Right, exactly. And you need a little bit of noise for deconvolution software in order for that to work properly as well. Um, So once you adjust your offset and your gain, and again, like we tend to use a low laser power and have our offset and gain between 700 to 800. I mean, obviously every scope is going to be different. Depends on how strong your signal is. If you have a really, really, really weak label and you really have to crank up your gain, that's also not very good because you can get some false signal in there. And if it's that weak of a signal, I just wouldn't use that segment or that neuron and try one that is much better. Um, there are some instances, especially in the Zeiss Zen Black software, where there's this auto brightness contrast correction tool, which I'll talk about little later on, and it really helps with optimizing the gain and offset. And then once you have everything set up, you want to set up your Z stack. And what's really important here is that you don't just, you see the start of your dendrite or your neuron, and then you focus through, and then you see where it just ends, and then that's where you're going to stop. And that's not, it's okay, but for our purposes, it's not, because we actually want to get a little bit of space above and below so that you're encompassing the entire um, neuron or the entire dendrite with space, because if you happen to say cut off the top of a spine or cut off some dendrites, you're going to lose that information, and that spine won't be good for quantification, or that neuron won't be good for quantification because you're missing data. So you really want to get a little bit above and below your
That's
specimen
also
of
especially
interest.
important for automated algorithms too. They need a little room to work, and so you need to make sure that you have a buffer above and below the section or the dendrite or the cell of interest, so that you've got enough space for the algorithm to determine where the branch is and where it's not, or where the spine is and where it's not.
It's a really important point that I think people need to really realize. And so here we just have a couple examples of some images that we took. Here's a low mag shot of a neuron that's filled with lucifer yellow, and there is some leakiness around it, but it's enough that we can still get around it when it comes to reconstruction. And in this instance, you can see how the soma is really bright and really saturated, and as well as the primary apical dendrite there. But then the distal dendrites are pretty faint. And so you want to adjust your offset and gain here so that, if you wanted to bring up your distal dendrites and increase your gain, your soma and your primary branch are going to be so intense you can actually lose a lot of important information. And when you think of it, this is just a 2D rendering of a 3D image, and your dendrites are not just coming out from the sides of the soma. They're coming out from the center of the soma. And there's a lot of instances where you have branching very soon after these cells, these dendrites come out. And if you supersaturate your soma, you're going to lose that. And you're going to lose information, and you're going to lose information on complexity. And so what you really need to make sure of is that you either take your neuron and image it in tiles and then montage that together, or you can adjust your settings accordingly. And that's why that autocorrect from available on the brightness correction on the Zeiss is a nice system. And if you look at the next slide, we can see, again, adding this false color filter, we tried to crank down the gain as much as we could in that soma. I mean, it's the brightest part of the cell. It is very difficult. And so here, again, we're setting it at two different settings. So we have one setting as we're going through the focal plane where we set the offset and gain. And here you can see again, when as we're moving through the cell, we're changing our offset and our gain so that we do actually have some red saturation, if you will, in the distal dendrites as we're going deeper and deeper into the cell. And again, it's really, you have to optimize that gain and offset. You don't want that super saturation because you will lose a
Right,
lot
and
of
so when you're
information.
looking at this live, all of these blue pixels and all of these red pixels are sort of dancing across the screen, kind of like a snowstorm. So they're not indicating that they're fully saturated. It's an indication of sort of noise, I guess, right?
Right. I call it the salt and pepper effect. And this is exactly what I was referring to. So here we just have a dendrite that we're looking at. And again, we can see two spines that are coming out of us in the Z plane. And if we had a supersaturation, we would lose those. And I mean, this image here, it's 2D rendering after we've imaged and after we've averaged as well. And so we've lost a little bit of that red that was actually in the original image. But again, you don't see a supersaturation across because you would lose those spines. And we just want the salt and pepper effect. We have a salt and pepper of the blue for the background and that sort of noise, and then a salt and pepper of the red the signal. And again, you can just see in the highlighted box the our settings for the gain and offset. And so we have a low laser power and our gain between 700 and 800. And like you really, and also keeping it at 1 ARRI unit, which is really important. And that's, there's a reason why the confocal microscope is set to 1 ARRI unit. If you open it larger, you're going to have too much signal coming in, too many photons coming in. And it depends. And then you have to ask yourself, is that signal really there or not? And for this purpose, you don't need to have more than 1 ARRI unit. It's different if you're doing a different immunohistochemistry sort of analyses. But for spines and for filled neurons, you really don't have to um, um, have more than 1 ARRI unit. So again, if we look um, again, imaging here, here's the spine we saw in the very beginning. We have the salt and pepper effect where we don't want to have too much saturation. If you go on here, we can look at now this auto correction. And this is nice, and this again on the Zen software, where if at the very top of the dendrite, it's really saturated, really strong signal, and the bottom is rather deep into the tissue, and you're going to lose your signal as you're going down, and the farther you get from the surface of the tissue. So you're really going to have to crank up your gain, and that can again have this super saturation effect at the very top of the dendrite. And so this Z brightness correction allows you to set an offset and gain at the very top and add it to the list or at this certain position. This is your offset and your gain. And as you move through the tissue, you can set multiple offset and gains. And then when it comes to imaging it, it'll save this. It'll save the offset and gain at each of these positions and then give you a nice image. And this way you don't have oversaturation in one side versus another side. And also what's important is sampling. And considering the size of a spine head and how small it can be, we do tend to oversample in our group. And people ask the question, what X and Y parameters do you use? What Z do you use? And for us, for spines in particular, we'll do a magnification of 100 with a 1.4 NA. We also will do a digital zoom of about 3.5. So then our final X and Y are 0.05 and our Z step is 0.1. And yeah, it's oversampling. You're always fighting with, the Nyquist. And considering though how small spines are, it's important, we do oversample just so we can get the resolution that we need. There is an optimal Z step people like microscopes will suggest. We always go a little bit smaller than that. Again, I think it really also depends on what your question is and what you really want to look at. If you're just looking at spine density and you don't want to type your spines, then you can maybe go with a bigger Z step. If you really want to type your spines and really be accurate about it, I would go with a bit of a smaller one. Um, and so again, I think the key points of imaging here is that we want to look, you have to determine what magnifications you want to use. What are you imaging? Is it cells? Is it spines? Is it spines on cells? Do you need to montage your image? And again, there's some systems now which really make it easy to tile, take multiple images and tile them together. And we've done that many times before, and we'll do about a 10 to 20% overlap when we're doing our tiling. And taking it at a 40x or a 63x resolution, in that neuron we showed about that we filtered by a site and after recording that was under 40x magnification. Our x and y were 0.1 and our z was 0.2. And again, and then even at this point we still got really nice spine identification, but I would not classify spines under those parameters. If I wanted to do that, I would go at a 63 or 100x resolution to do that. Um, and again, as I mentioned, optimizing your offset and your gain, you want to avoid photo bleaching because that would really suck if you bleached out your entire neuron with this, one-time imaging. Because sometimes you do want to go back to them, and look for other, get a better magnification of your spines or get the picture for your publication. Um, as you mentioned, capturing images capture image planes above and below your entire cellular dendrite. And every confocal will be able to do this, whether it's a Zeiss, Olympus, Nikon, Biorad, Leica. Um, spinning disk and the other sort of mock systems can do this. It'll take a much longer time to do this. I'm not sure on the resolution. I don't think it's as good as a confocal, but I've never really had that much experience with it. You may have to fiddle with the system to see exactly, what sort of resolution you can get. But I think it would just take much longer and to obtain the image versus a conventional
Yeah,
confocal
I think that
microscope.
there are people who are using spinning disk and other structured illumination
Hmm.
scopes for applications like clarity, where you have this entire large tissue volume that you want to be able to image through, and being able to capture the frame at once rather than build an image over time. There's a trade-off, and you can optimize things not just for the resolution you need for the quantification, but also um, divvy up the how much attended time it takes at each of these steps. So maybe you can set up your microscope using Neurolucida to capture those image stacks for montaging and have it stitch automatically for you. Um, and so that setup time may be 10 or 15 minutes, but it acquires overnight. And that sets you up so that you can do other things as well. The same thing goes for what type of confocal you're going to be, or what type of microscope you're going to be using and controlling. So, Neurolucida controls some Zeiss confocals as well as some other confocals as well. So there's lots of different considerations for how you acquire your tissue. The ability to take that montage frees you from being stuck to a single field of view, so that the automated tools have really come to the forefront here to really allow you to capture more and more and more image data and stitch it. So even if it's just 4 or 5 or 6 tiles or 11 tiles, in the case of that neuron that you were talking about before, or hundreds of tiles when you're talking about a clarity specimen, the the technology is advancing rapidly, and so now the volume of image data that you have to quantify from is just increasing exponentially. And so it has an important part to play in all aspects of your experimental design, for sure. Oh, so speaking of, this is a clarity specimen, GFP, transgenic mouse. I think this is a cerebellum, and it just goes to show that you can have quite a volume that you can generate data from, and now the question becomes, how do you quantify it effectively? Let's pause for a couple of questions. There's been a lot of questions that have been submitted, and I think that that's absolutely fantastic. Please keep them coming. I know that we've got a lot to say and a lot to show you, so we're going to keep some of these questions relatively brief and then move on to the next topic. But the questions will be answered and after the webinar as well. So let's see. Oh, here's a question from Ms. Valera. The question is, once you have all your samples and your labeled neurons, how do you get the same orientation of the neurons in different specimens to do a proper comparison? So at that point, that's an analysis question, and I'll make a mental note to show you how to do that in Neurolucida. It's really quite straightforward to be able to rotate the image volumes just so that you can have that defined, apical is up perspective, even though you may have been recording or imaging from a neuron that is oriented in a different perspective. Angela, Dara, has a quick question about how you injected the fluorescent dye for the cell loading. So what did you use? Did you use a pico spritzer or uh, um,
We use a glass
okay.
micropipette. And so you can incubate the tissue in DAPI to figure out where you are in your brain section. Um, if you're looking in a specific layer of the neocortex, like layer 2, 3, or if you want the pyramidal layer of the hippocampus, CA1 or something. So we use DAPI, and then under the DAPI filter, we'll focus in on a cell completely random and then just inject. But some people also will just do blind injections, just under a fluorescent filter and just kind of poke the tissue until they hit a cell body and then just start filling that way.
That seems pretty magical.
And it's
Yeah,
under a current as well. So as soon as you kind of put your pipette into a cell, you turn on your current and the dye just kind of rushes on.
got it. Okay, well, let's move on to quantification because some of the questions that are coming in right now have to do with once you've got your images, now where do you go from there? So let's not delay. Let's move right on to quantification. So before you do that, however, if you're analyzing spines, you might want to do one more step between image capture and analysis, and that's a pre-processing step called deconvolution. So Dara, what can you tell us about this?
With the deconvolution, as you can see here, this is just an example of one of our dendrites, and um, it's an imaging processing technique that improves the contrast and resolution of your sample. And we use the blind deconvolution algorithm. This algorithm estimates the point spread function from the image by calculating the blurring in all three dimensions and then compensating for it for other optical aberrations. And so as you can see, it really makes it gets kind of any kind of um, unfocused pixel or voxel, and it really kind of makes everything much more crisp. Um, and this could be important in that, in terms of spines, you can have a spine which unprocessed might be considered a mushroom, but once you actually do run it through the deconvolution, it actually is a thin, or a stubby. The neck can also all of a sudden be resolved once you run it through a deconvolution, so now it's a mushroom. So it's really important to run it through such um, um, software, and it also will help get rid of any sort of Z smear or any other sort of aberrations that you acquired while
It
you're
doesn't work so
imaging.
well when you've got sort of a head detector problem with your confocal. So you get those line scan issues that can crop up with confocals. So you need to make sure your scope is in good working order when you capture your images as well. But deconvolution is a great tool to help to create that digital image in as clear a focus as possible that represents the way that the tissue was um, in its original state. And so you're trying to identify the point of origin for each of these photons and then remove any extraneous noise that is a consequence of the tissue around it, the mounting media, the cover glass, and all of the other factors that you're trying to subtract all of those features away using these mathematical models. So now that you've got your nice, clean, crisp, clear, deconvolved image data, tell us how you quantified.
Well, when I first started, we did it all manually. And so we would do live tracing of neurons using Neurolucida. And at first it took a long time, but as we got better, it would still take about 45 minutes to an hour to reconstruct a pyramidal neuron from the CA1 of the hippocampus. And it was many, many hours in a dark room and hunched shoulders. And I would go to sleep, close my eyes, and I would just see green neurons everywhere. Um, and then to do the spine quantification, it was all manual as well. So you would upload, the stack into Neurolucida and then go through the Z, counting them one at a time. That was just counting; it wasn't even classifying. And so, classifying was more difficult. And there's a few paper from John Morrison's group, which we just highlighted here, where they actually went on Photoshop and did manual measurements of thousands and thousands of spines, looking at head diameter, neck length, neck diameter, um, head volume, and it's a lot of painstaking work. But then soon after I came to Mount Sinai, the Neuron Studio came along, and this was an automated way of quantifying spines and even neuron structure. But here, as you can see in panel A, this 2D image, there's arrows going 1, 2, and 3, which are actually spines, which we don't see looking at it just straight on. Where in reality, there are spines, as you can see in B and C. They're in B and then colored out in C. And even though we can still see these, we don't know what type they are because they're still coming out at us. Are they stubby? Are they mushroom? Do they have a neck? It's kind of really difficult to see. And the beauty about Neuron Studio is that it quantified in 3 dimensions, and you can see in panel D where they turned it. Um, you know, we can see a possible mushroom spine there and a stubby spine there, and having that ability to quantify it an automated way saved on so much time, and it was much more accurate than the manual counting and manual classification that we had to do previous to that. So it was a godsend, pretty much.
Well, not too long ago, Neuron Studio was no longer able to be developed. The resources weren't there to continue its development in the academic realm, and there was a program by the National Institute of Mental Health to take information, these techniques that are being developed in academic labs, and partner with commercial organizations to keep this technology that is of use to many researchers and expand it and make it more robust and further development. And so Neurolucida 360 is actually a research grant that we received from NIMH together with you folks at Mount Sinai, Patrick Hoff, and you. And the goal of this is to commercialize and further develop the technology, not just within Neuron Studio, but to improve the tools that already exist in Neurolucida. The automated reconstruction tool AutoNeuron, as well as AutoSpine, and even some of the pre-steps that are important, like image montaging, to be able to collect and create these image volumes that represent the entire structure that you're interested in quantifying from in a way that can allow you to automatically reconstruct and extract that those metrics that you need in order to make statements about your hypotheses. So Neurolucida 360 is the result of all of this hard work. It's a grant that is still ongoing, so we've got lots of work that we're continuing to do. And while we're focused right now on fluorescently labeled material, we also work in bright field, 2D and 3D images. So if you're working in vitro preparations, the Neurolucida 360 will analyze that data as well. From fluorescent material, it can be created with a confocal or multiphoton, light sheet microscopy, or even standard epifluorescence. Once you start talking about light sheet, you get into the big realm of clarity and other big data. So again, as you start talking about networks and larger data volumes, the automated tools, in order for quantification, need to be scaled up in order to be able to manage that large, complex image data, gigabytes, terabytes of data. And NL360 is designed to do that. Golgi impregnation is an area of active research for us. I'll tell you a little bit more about it, where we're at, and what makes it sort of a special case scenario. But likewise, Brainbow has additional and interesting challenges. So even though it's a fluorescent method, the fact that each cell is labeled with multiple fluorescent fluorophores indicates that it becomes a question of hue rather than straight up color that defines what a neuron is. And so all of those things represent areas that are extraordinarily complex, but represent where science is going. And so, of course, Neurolucida 360 is going to take you there. We also work with high bit depth images. So if you are working with a camera, or after deconvolution, you've got 16-bit image data, you no longer have to downsample that to analyze it. We work with that, as well as a wide variety of image file formats. What this is intended to show you is that Neurolucida 360 is taking the best of Neuron Studio and the best of AutoNeuron and the best of AutoSpine, and then adding a bunch to it to create this new 3D interactive tracing environment for data collection. And so that's what I'm going to show you right now. So I'm going to bring up Neurolucida, and I'm going to bring in a couple of image files from a customer by the name of Rebecca Piskarowski. Thank you very much for providing me access to these images. They're lovely. So we're going to load these in a grid, and I'm just going to quickly demonstrate this microscope-centric view, which is an XY view where I'm paging up and down through the stack. So I'm stepping through the individual focal planes by just going up and down in Z. And you can see that this is a very microscope-centric view, and this is how Neurolucida has been designed to work from its inception, because it was originally working live at the microscope, and now, of course, we allow you the capability to quantify right from image data. But now with Neurolucida 360, we're going to take it out of this XY-centric view and allow you to collect all your data in this 3D interactive environment. First, however, we have to stitch this montage together. So you can kind of see here, there's this hole in the tissue here. These two guys, that's where they overlap. So let's take a look at that. So maximum intensity projection, you can see that this is a gorgeous cell, but it's not too gorgeous. This gets back to what you were saying, Dara, right? About pretty, but not too pretty.
Yeah, exactly.
So I'm going to go into image montage mode and stitch it. So all I did was hit the automatically align images and blammo. There you go. It's stitched, and it's there's probably, I don't know, 10% overlap here. And the amount of overlap that is needed when you're creating these tiles for montaging, it depends on how much image data actually spans both tiles. So if you're very distal and you've only got a few structures, or you're trying to capture the axon and it's a very fine process, you're going to want more overlap. But when you're right in the thick of things, this apical dendrite or even the basolateral dendrites, there's a lot of material here for fiducials. So a lesser degree of overlap is perfectly acceptable. So the next step would then be to write out that image file. As instead of two independent files, we're just going to create one. But to save some time, I'm going to throw these guys out and bring in the stitched file. And so I'm not sure how this neuron was created. Maybe Rebecca can pop in, and I think she's attending the webinar today, and if she wants to chime in and tell us how this neuron was visualized, that would be great. But so we've got our stitched montage, and so if I step out of maximum intensity projection, I can walk through the stack, and so you can see that it's a nice seamless stitch. But now I'm going to bring up Neurolucida 360. So this is this new environment that we're very, very, very excited about. And so you can now easily interact with your image volume, and not just look at it, but actually collect data from it. And so I'm able to rotate the image volume just holding down the left mouse button and moving my mouse around. You can zoom in and out using the scroll wheel. And I can, on the right side here are all these different panels that allow us to actually create data. And so I'm going to detect the soma and start reconstructing some of these branches. And so I'm going to zoom in a little bit. And so we have multiple modes. You've got manual, interactive, and fully automatic. The fully automatic is still in development right now. It's almost ready for prime time, but right now, not quite yet. I'm going to spend most of my time today working with interactive because it's a nice mix between the manual and the automated, because it uses automated algorithms. In fact, you have 3 choices. If you are an AutoNeuron user, you may be familiar with the directional kernels algorithm. This is the engine that AutoNeuron runs on. If you are a Neuron Studio user, you may be familiar with voxel scooping. And as a result of this grant from the NIMH, we've been able to develop a 3rd algorithm called reverse crawl. What these algorithms are just different methods for distinguishing your object of interest, so the dendrite, from background. And so what we can do is just start tracing. So I'm going to pan to window center after each click. And so this is interactive, and so we can walk up each branch. We can zoom in as we're going to get a better feel for these tight corners to figure out how it should work. If you make a mistake, just a Control Z sets you back. If you want to work on a different branch, you can trace in any direction. It doesn't have to be from the soma out. For those of you who are used to using Neurolucida or tracing live from the microscope, you do have that sort of soma-centric view because that's where the party gets started. So just switching to this 3D environment really allows you to view where the automated algorithm is able to distinguish what's going on. And I really find that extraordinarily fascinating. I could do this all day long. So let me just, before I get too bogged down in the fun of just tracing, it'll automatically connect branches for you, so you don't actually have to do anything special to do it. So as I'm approaching the segment here that already exists, you can see that my guide has turned red, and the point of attachment is this teal sphere. So when I click it, my green branch is now going to turn red, indicating that it belongs to that branch. And so we can create these structures very quickly, very easily. You can switch between the algorithms at will. You don't have to reconstruct using the same method, and those methods are really just designed to give a different amount of weight to determining what's signal and what's noise. So what's the branch and what's not. And you'll find that the different algorithms perform differently on the different types of image data. So rayburst crawl I prefer for di labeled material. Directional kernels works great for the discontinuous labeling, so where you've got some breaks and some stutters. Directional kernels is really powerful for that. So you can keep doing this reconstruction, and then one other little segment here that we can do is we can create a quick view for your publication. So like Dara was talking about, you can, when you're creating your image for the cover of the journal that you're submitting to, you can do a quick post-processing on your model to unify the image and make it look a little bit better for display purposes. When you're going back to reconstructing the image, it goes back to displaying the data on a branch-by-branch basis. There's a lot of interactive editing tools which are really pretty fabulous. And so you can move points in Z, X, Y, and Z very easily. You can delete them. You can change the diameter and the thickness at will. You can also detach and connect. So when you've got these branches where they're really dense, you can trace them and then come in here and edit them. And this is something that is a big advantage over existing tools. This 3D editing is really very, very powerful. I really think it's just super helpful for visualizing what's going on and making things quick and easy for that quantification process.
Susan, how different is this interactive version versus the
works the
completely
same way.
automated
They
one?
use the same algorithms. It's just when you are using the interactive mode, you are providing a bit of guidance for which segments belong to which branches and how these complicated connection points should be connected. So when you've got a real dense area like this, where that Z information, that axial resolution just isn't quite there, interactive tracing may be something that you might want to switch back and forth between. So you can even do manual, interactive, and fully automated all together, just hopping between the different methods for the different parts of the image. So they use the same algorithms to answer your question. Okay, so this is the structures for tracing the somas and for interactively tracing. Let me switch and show you what spines look like. So in this case, this image data isn't the resolution necessary to pick up spines, so we'll actually demo that using one of your image stacks. Thank you very much for sharing that with me. So I'm going to load that real quick. And so this is a deconvolved stack. What cell type is this, Dara?
This is a dendritic segment from an apical dendrite from a CA1 pyramidal neuron in a mouse.
So we're going to trace the dendritic backbone. And then I like to inspect the dendritic backbone before I detect the spines to make sure that it's really accurate, because when you're imaging at this high resolution, these spines are actually relatively large in comparison to other magnifications, and there are a number of pixels now that make up that spine, and so the algorithms can get sort of what's that called? Distributed or pulled by the weight of the spines. And so I like to ensure that I'm looking at the right, that they're not unduly influenced by the structure of the spines, so the presence of the spines. And so I might do a little fine tuning here to make sure that the spines really, the dendrite modeling represents the dendrite before we do the spine detection. So you can represent the spines or the dendritic structure in a number of different ways. You can turn on and off the tracing just to ensure that the dendrite is modeled for thickness appropriately. You can also display the dendrite in centerline mode, which is pretty handy, I think, when it comes time to reconstruct the spines. So let's do that now. The spine detection algorithm that is utilized for modeling spines is the engine that was originally developed in Neuron Studio but has been further enhanced in Neurolucida 360. There are a number of settings that you can specify to fine-tune the detection. The first is the outer range, and so that is the distance from the surface of the dendrite out. The minimum height is from the surface of the dendrite model, so the tracing of the dendrite relative to the pixel data that is behind it or underneath it. And so this is important for distinguishing between stubby spines and, say, the dendritic surface. Your detector sensitivity is set at 100% initially, but you can go above and below 100%, and that just allows you to pick up more faint signal. And so you can modify that as necessary. And then the minimum count is, as you might expect, the minimum size for the detections. And so once you have settings, you can click to detect, which is very gratifying. It's very, Zen-like. I could spend a lot of time just sort of clicking and seeing what happens. And so here's our dendritic spines that I've modeled just by clicking in the image. You can also let the software do all the work for you and do find all. Each of these spines, the different colors actually don't mean anything right now. They just indicate individual spines. But if you want to classify the spines, you can select this button here, which is Classify All. This is based on the method that is integrated into Neuron Studio and was validated in the Rodriguez et al. 2008 paper that Dara referenced earlier. And so we've incorporated that. So with a simple click of the button, you can see which spines are which. Red is thin, green is stubby, blue is mushroom. And you can come in also and edit these spines too if you want. So let me see if I can find a good one. So let's take a look at this guy. This one looks like it's two spines, but let's say for the sake of argument that we feel it should be one. So you can select both of those objects and merge those spines, and then even change its classification. So you can make it a mushroom spine if you'd like, or any of these other options as well. You can change how they're displayed. So just like with the dendrite, you can change how it's modeled on the screen. And you can also do a marquee selection. So you can select multiple spines and remove them or change them. And so in a quick and easy manner, you can collect quite a bit of great data. Why don't we pause for questions? Dara, tell me about floating spine heads. Like, that's because there's not enough fluorescence in the neck because the neck is so thin, right? So that they appear to
Yeah.
float. What do you do in that instance? I mean, it's obviously still a spine.
I mean, in the past, we've counted them. Like they're thin spines in our experience, or just the spine neck is just so thin. It just doesn't. It's below the resolution of the microscope that you can
Hmm.
get, right? But it's still considered a spine.
Well,
We
yeah,
consider it a
because you're
spine.
able to calculate the head to neck ratio, and if the neck is so thin that it approaches, 0, then it's still very
Hmm.
thin. So
Yeah.
that you're able to model it and classify it on the basis of that. Another question that has come in is whether or not we can do shoal analysis, and the answer is yes. Neurolucida has done shoal analysis for 25 years, and so it is a simple analysis request in Neurolucida Explorer, which I'm going to talk about yet. Tyler asks about manually setting the parameters for the spine classifications, and that's an upcoming feature. I think that that's really great to lead into because we've established the classification that is currently integrated into Neurolucida 360 on the basis of the Rodriguez et al. Paper, um, but we recognize that that may not be how you want to classify spines because of a different disease state or a different species. And so we will integrate individual user classified classification schemes in the software. It's coming up very shortly. As it is, the analyses that are generated in Neurolucida Explorer from the Neurolucida 360 data allow you to classify according to the tree and the branch order. So if you've got one of those montage neurons where you've got the spines, you can calculate the spine density, the number of spines, the distance from the soma, and then also all of these classifications as well. So if you've done an automatic classification so that you've been assigned a type, you'll see that it says auto. In this instance, I selected that spine and changed the classification. So I changed that one to other. And so you can see here that that was a manual choice that I made. And then all the way across, there's about 25 metrics that are provided for each spine that include everything from the total length of the spine, so from the surface of the dendrite all the way out to the head, from the surface of from the head all the way to the center of the dendrite. So if you're interested in spine characteristics based on the size of the dendrite that they're attached to, you can perform those analyses. You can also look at things like volume, the connection surface area, as well as the angle of emergence from the dendrite. So if you want to restrict your analyses to spines that appear in plane to the XY view, so if you're looking at any other bright field imaging where you want to restrict the analyses to spines that you can clearly see, you can do that.
I think one important point to make is when you are doing manual classification, if you're going to change the classifications of spines, there's a lot of work in EM which have already established what parameters spines are, and that should really be taken into consideration when you are going to change it for you're changing the parameters, because otherwise, if you're looking at a specific disease state, you can easily bias your data, and you should really kind of go based on specific spine sizes that have been already established through EM quantification and for specific brain regions and for specific species, because otherwise you can just kind of randomly take out of a hat and, you know, bias your data, which could be true or not true. So I think that is something that really has to be considered.
Yeah, I think you're right. I mean, the place to go for the classification metrics is the primary literature, and we're gonna model that as best as we can, for sure. Ben has a question relating to montaging images, and I think that this is really important because it used to be very difficult to handle multiple image stacks, but the Neurolucida, we've spent a lot of time creating algorithms that allow you to automatically stitch those stacks in all 3 dimensions. So the XY as well as within Z. The tool that I demonstrated, so this image montage that I started off with, I showed you the maximum intensity projection so that you could see the full extent of the image data within those stacks. But the algorithms themselves actually work in the 3-dimensional space. So even though we're viewing the maximum intensity projection, all the algorithms are working with the entirety of the image data. And that's true also for all the tracing algorithms too. So the spine detection as well as the branch tracing too. So let's move on to Golgi. There's a lot of people who have been patiently waiting for us to address Golgi. And Golgi is just a wonderful technique. It's a technique that has been around for well over 100 years, and it provides some stunning neuroanatomy. The neurons that are filled entirely from their axon all the way to the fine resolution of their dendritic spines, and it fills each of those cells completely. So it's a very powerful technique, but it has some complexities that make it difficult to analyze. With Golgi, you have no control over which cells get labeled and how many in a particular area get labeled. And so you can have this high density of labeled cells, which can make it more difficult to reconstruct manually or even using automated methods. You also have commonly these dendritic breaks where these segments get broken in the process of the labeling, and assigning those breaks to the different neurons can be complicated. The final thing that makes Golgi hard to do, but is common also to other bright field imaging modalities, is discerning spines that are coming out at you in the image plane. So because the light is coming from below, because it's a bright field imaging, the light is coming up from below. It's being sent to the camera through the objective lens, and you're obscuring the presence or the ability to see these spines based on what's above and below them. And so it could be another branch, it could be its own dendritic branch. And so the ability to determine and model spines in these areas of obscurity, so above and below these dendritic planes, so how they're being obscured, can make it very difficult for you to quantify either manually or automatically. It's an area of intense research here at MBF. We recognize the power of Golgi and have been working very hard at coming up with automated methods that allow us to extract more and more information from this imaging method or labeling method. And so I just want to show you briefly the status of our research. So right now, you're familiar with Golgi, and if you invert the image, you can really see the impact of the precipitate that forms in the process of doing the Golgi, and just how branches above and below where you're looking can obscure the image data near it. So the dendrites and spines that are next to it. And so the challenge is to come up with a way to identify what is a dendrite, so what is a neuronal structure and what isn't, and try to extract the neuronal image data or retain it so that we can filter out the blur and try to extract that. And so we've made some great progress, I think. And so this particular Golgi filter is in development, and I think we're getting there. And so I'm really pretty excited about this. Alfredo Rodriguez has been working very, very hard on this, and so we're really, really thrilled with his progress. And so once you've employed this filter, you can now use those automated tracing algorithms to model not only the branches but the spines themselves. So as the software exists right now, Neurolucida 360, you can trace Golgi, dendrites, somas. It will vary depending on how complex the image scene is. So your success will vary depending on how complex it is. But we can utilize Neurolucida 360's tracing algorithms to really extract that information. Spines are complicated, but I think we're making good progress on it. To summarize, for automated reconstruction, the quality of the data that you're going to get out really depends on the quality of the data that you're putting in. So it really depends right at the inception of your experiment to really think about what sort of analyses you want to do and to optimize your entire experiment for those analyses. And that starts right at the cell visualization. So Dara spent just a wonderful in-depth conversation on how to prepare material for fluorescence labeling and imaging. And so she went over some of the common methods for visualizing cells using fluorescent methods, and then how and what to do when it comes time to think about imaging. So the resolution, the XYZ scaling, how you consider image montaging when it's appropriate, as well as post-processing techniques that can be utilized to even further improve and disambiguate the image data of the neuronal structure that you're interested in. And then I introduced you to Neurolucida 360. It is our interactive 3D environment for data collection, and it provides a number of algorithms for manual, interactive, and fully automated tracing of somas, dendrites, and spines, synapses. And it is in active development, and we are very much enjoying all of the fun research that we can do to improve the visualization, to expand to bigger and bigger and bigger image data sizes. So working with clarity data sets that are terabytes in size, it's pretty fascinating. Because of that, the computer hardware really matters. So Neurolucida 360 runs in a 64-bit environment on Windows operating system. The more powerful your PC, the better. In addition, it's very graphics intensive, and so you need a nice, powerful graphics card in order to really utilize the full power of Neurolucida 360. So when you utilize NL360, you just want a nice screaming machine. The bigger, the better. And then as is the case with Neurolucida, Neurolucida 360 has just an extraordinary number of data analyses that have been integrated into Neurolucida for the past 25 years, and we're going to continue to add even more specialized analyses as the methods dictate that. So everything from the spine modeling that we're doing in Neurolucida 360 to different ways of incorporating the network analyses that you may be interested in. So it's a wide variety and very robust and customizable to your particular research question. If you want Neurolucida 360, contact us. You can get a free trial by going to this website, mbfbioscience.com, Neurolucida 360 free trial. You can email us at info@mbfbioscience and go ahead and give us a call. We're happy to talk to you. You don't have to do it all electronically. We'd be happy to chat with you. There's more information on our website. And even though this is Dara's first webinar, I think she really hit it out of the park. Dara, how do you feel?
I feel good. Relaxed now.
I think you did a great job. I've done a few webinars, and MBF staff has also, I'm just one part of the webinar group here, and all of our webinars are available to watch after the fact, including this one. You can see them by going to mbfbioscience.com webinars. We also keep track of people who publish with our equipment and software. So when you publish, let us know. We'd like to cite you on our website. So primary literature references are available on our website. And then the methods that we reference and use within our software are available in the context-sensitive help so that you can see exactly where we're coming up with these strategies for evaluating your data. We don't keep anything hidden. So I hope that even though we've gone long, I really appreciate your attention today. I'm happy to stay online for a little bit longer if anybody wants to ask some questions. I know that we didn't have a whole lot of time because we had so much to show you, but I really appreciate your attention. I hope you'll contact us for a free trial and have us introduce you to Neurolucida 360. It's the interactive 3D platform for reconstructing bright field image data, fluorescent image data, confocal, clarity, Golgi, spines, axons, the full network. So thank you again for your attention. And thank you, Dara. I really appreciate you doing this with me.
Well, thank you for asking me to do it with you. I really enjoyed myself and all the questions that people are asking, and I'm trying to answer them as we're going. It's taking a little while. So
it really has been a really popular topic. There's been just a ton of questions. Oh, Ruby asks if there's a way to do this quantification away from the physical microscope. Can I run Neurolucida on my laptop while I use the scope to capture more images? Ruby, the answer to that is absolutely yes. So Neurolucida 360 is untethered from your microscope because you're bringing in image data into it. So if your laptop is a nice, powerful laptop with a strong processor and a good graphics card, you by all means can use Neurolucida 360 on your laptop away from your microscope. You could do it at the beach. You could do it in New Orleans, anywhere you want it to be. It doesn't have to be in the dark, cold scope room. You could be sipping a cocktail if you wanted. Eric asks about how the program can be beneficial for whole brain quantification, in parentheses, clarity, for tracing white matter tracts through the whole mouse brain. Eric, it's a very important direction that neuroscience is moving towards, the sort of, I'm going to call it holistic, but the gestalt of the brain. Now that we're able to generate these extraordinarily large image data sets, we need the tools to extract quantitative metrics from them. And so Neurolucida 360 will be capable of operating on these extremely large image data sets. We are working with them now, and we are continuing to add more tools that allow people to image and analyze subsets of data in ways that make sense to them. So we've got a couple ways to do it right now, but part of the process of this development is making the tools very extensible for the different ways that people like to work. So sort of a user experience sort of perspective. And definitely contact us, and we'll see how I can show you that our software works for your clarity tissue. I can hear Dara typing in the background. I think she's answering a bunch of questions.
Yeah, sorry, I'm just
Yeah.
answering a
Oh,
question
that's
for
great.
someone.
What question is it? Bring it to the conversation here.
It's a question by Jill about a technical question for tissue processing, whether the concentration of glutaraldehyde that we use has an issue with autofluorescence. Um, and if they have to get rid of the autofluorescence for successful image acquisition. And we don't, with the amount of glutaraldehyde we use, we don't really see any issue. We don't have any issue with autofluorescence. I think you would have more of an issue with lipofuscin in this situation over, the autofluorescence from the glutaraldehyde that we use.
So, how do you? Speaking of lipofuscin, how do you minimize that? You must have to deal with that in aging.
Yeah, we do have a lot of that in our aging animals, and especially with the Alzheimer's disease mouse models. But there's a couple ways. There's a really good review or a paper by Shell et al. From 1999, which kind of went through what's the best methods for getting rid of the lipofuscin or reducing it, and pretty much just kind of incubating it with a solution of Sudan black B or in a solution of copper sulfate. Um, really helps get rid of this lipofuscin, and it can make the tissue a little bit more fragile. But if you're using thick tissue sections, it's not, I don't think it's as bad as if you're going to be using, like a 50 micron um, thick section like you would for traditional immunohistochemistry. So, be careful, but, you can always stick it in there um, if need be. And I think, again, we're focusing on more large structures and neurons. Some of the autofluorescence I think would have more of an issue when it comes to like if you're going to be staining for puncta. But I think in this situation, the minimal autofluorescence can be really overcome by, changing some of your settings when you're imaging or through the deconvolution
Mm.
process.
Do you think that some of the mounting media choices have an impact on autofluorescence, or not so much? Because I know like Movio continues to sort of clear the tissue the longer it sits in it, and I wonder if that would help abrogate some of the autofluorescence.
I've never used that. Um, I don't know. I know with our mounting media, we don't really have such a
Mm.
huge issue. Um, there's some that have the anti-fade, which I think are
Oh,
great
right.
and help keep the signal a little bit longer. But I've gone to neurons that I loaded, I don't know, 3, 4 years ago, and I can still bring them out and image them. I mean, they faded, but we are still able to see them, and that's from a few years ago. So that's just shows it can last
Well, I
for
think
quite a
that
while.
also shows that you've got a stellar technique and you're very proficient at your job, that you can, so cleanly label your neurons and fill them to just the right amount, not too much, not too little, just right, and that it can retain that label for that long. I think that's really cool. Marcel wants to know where you get your spacers again. Can you give those details again?
Yes, so we get them from EMS or Fisher. Um, I can send him the information. I can get the catalog number for him, and I can get that later. I don't have it off the top of
Okay,
my
we'll
head.
post that on the website because we're going to answer these questions. So we'll provide that information um, there on the after the webinar. That sounds great.
Oh, Scott just came in saying there's another way to deal with lipofuscin is to use a spectral detection system,
Oh,
like a Meta or Quandra, and online unmixing when you acquire images.
thanks, Scott.
So it's easy. Yeah, that's a great. I forgot about that parameter on the systems that you can do that. So that's really helpful too.
Yeah,
Thank you.
yeah, because it's a broad spectrum. Uh, it shows up in many
Yeah.
wavelengths that you can
Yeah.
unmask it that way. Haley asks, what's the advantage of using Neurolucida over ImageJ or Neuron Studio other than convenience and ease of use? I have a vested interest in this because, of course, I care greatly about Neurolucida. I also care greatly about convenience and ease of use, so I think that those things are extraordinarily important. And I'll let Dara provide her feedback too, because she's spent many years using Neuron Studio since she has been you were very important in the implementation of it. But Neurolucida has a few things that are really important, I think. One is continual development. This is our full-time job. We are not also sitting in the lab filling cells and doing other things. This is what I do all day. I talk to scientists like you, figure out what your research questions and problems are, and convey those to our full staff of programmers who make that dream a reality. The other thing that Neurolucida has going for it, similar to the other programs, but there's a long lifetime of development that has been invested into it, and so you have 25 years of analyses, like I talked about for Neurolucida Explorer, that go run the gamut from everything from shoal analysis to convex hull to individual spine density questions to classification to fractal analysis. There's just an extraordinary amount of analyses that you can pull out from your data that you can go back to and enrich time after time after time. So if you've got one of those large montages of your neuron, you can analyze it in bits and parts and keep coming back to it and mining it for more and more information. So maybe you've got that beautiful neuron that Dara showed with that self-fill. Initially, she may just be interested in measuring the dendritic length to see if that animal, that cell from that animal in that group, has a decreased apical dendrite length or something like that. But she may go back and look at branch points or branch angle analysis. Come back and look at the spines and whether or not the density of mushroom spines changes depending on the distance from the soma. Like the way that you can expand your analyses, I think, is one of an extraordinary advantage of Neurolucida. Dara, do you want to add anything to that?
No, I mean, I think there's over 100 analyses you can do with Neurolucida. Is that what you mentioned the other day?
There's tons. Well, the other thing, actually, you know what? You reconstruct your neurons in Neurolucida live and then take your shoal analysis over to the confocal and say, okay, now I want to image at these particular locations.
Yes.
I think that ability to mix modalities between live and imaging and quantification is something that is impossible with ImageJ or Neuron Studio because they don't control microscopes. And so that right there, I think, is a strong advantage for Neurolucida users, especially because we control so many different types of microscopes. Everything from your bright field, simple non-motorized scope to, confocals. So there's a lot of flexibility there. You guys have been so attentive. I can't believe how many people are still online listening to us banter. How about we do just a couple more questions and then invite everyone to take this conversation to Twitter. I don't know how many of you guys are on Twitter, but that may be something fun that we can do. Scott popped on and gave a gave a reference for unmixing for lipofusion. Thank you, Scott. It's an article from Susan Weirn. So that's great. We'll definitely put that on the website so that everyone can have access to that, as well as the citations that Dara listed when she was speaking as well. So we'll make those in a nice, convenient location so everyone can see those. Oh, Scott says shameless self-promoting. He's a co-author. That's great. I love it. Well, that's okay. So I'm not the only one who's a little biased. I'm a little biased about Neurolucida, and you've got some great reasons to speak cogently about lipofusion. That's great. Okay, let's end up with this one, Dara, if you don't mind. Can you answer Adam's question? He wants to know if there's an advantage to perfusing the animal with PBS in order to flush out the blood in the brain before staining the tissue. He's using Golgi. And he found, or a recent study identified that fixatives normally used in perfusion caused glial cells to be stained rather than neurons. But if only a PBS solution is used, the neurons were stained clearly without glial cells.
Interesting. I mean, for us, since we're not doing a stain, we're just doing um, injection. Um, we don't have that problem of glial cell staining, and we found, I mean, this protocol that I mentioned for the cellular injection was optimized many years before I entered the lab here at Mount Sinai, and um, you know, through John Morrison and Patrick Hoff's lab here, who kind of established the cell loading in the 90s or even before, in their labs, they found the best um, fills where when they did a perfusion with a 1%, which is not a very strong fix,
Mm,
when they did the flush with the 1% and then followed it up with the 4% with the glutaraldehyde. So I know for all of my other immunohistochemistry techniques that I do a flush with a PBS followed with paraformaldehyde. But in these situations, I do the 1% for the injections. I do the 1% followed by 4% and
mm.
glutaraldehyde.
So one last thing, I guess Marcel and Lynette would like to know a little bit more about what you do for your cell fills. Marcel wants to know about what streptavidin dye you prefer, and Lynette wants to know what magnification you use.
So when we do, I'll answer Lynette first. When we do the cellular injections, you have to use a fixed stage microscope. If your stage is moving, you're going to break your tip every time. So we want a fixed stage microscope where your objectives move up and down. And depending on the tissue of the species. And so what I usually do is I'll locate my region of interest on a 10x objective. And I use a water immersion system. There are other systems which are not, but I like to have the water immersion. I feel I can get the best optics
Mm,
that way, um, as opposed to a regular air objective. And I'll find my region of interest with a 10x objective, and then I'll move to, when I do my mouse work, I'll move to a 40x, a 20x or 40x. If I'm injecting a monkey, I'll use a lower magnification because the cells are much bigger and I want to see the distal tips filling. So I can move to a 20x in that situation. And even then, I still don't see, it's hard to see all the distal tips filling, sorry. Um, but it depends the region you're looking at. It depends what cells you're looking at, and it can change. What's important is that you really want to be able to see the cell body. I mean, if you're using DAPI as a guide, just so you can get an accurate injection of your tip into that cell, and so you don't get too much leakage around your region. For the a question of the streptavidin, our collaborators, when we did all this cell filling and recording, they used the streptavidin Alexa 546 from Vector Labs at a 1 in 500 dilution. Um, and that's what they've been using for all of these experiments. And again, they also get their biocytin from Sigma. So anything that's kind of,
pretty standard,
the
yeah.
usual. Yeah, pretty
Please
standard.
contact us for a free trial. If you've got further questions you want to bring up or add to the discussion, I'd love to hear from you. And I'll be at Neuroscience this year and invite you to come check that out. That's a few months away, but I'm still thinking about how I just submitted my abstract. So I hope that if you are in Chicago, you do stop by and say hi. So, Dara, thanks again. It's been a lot of fun.
Thank everyone for
Okay,
for listening.
I'll talk to you later, guys.